in case of prostate cancer, so surgery is the we can remove the prostate cancer completely using the surgery, or still we have to give some chemotherapy?
So what happens in prostate cancer? Prostate cancer, thankfully, is caught at an early stage. And why? Because usually when people, some younger people also get it, but people are more prone to get it when they are older. So there are some guidelines in US that if the person is above certain age, I don't know what age, maybe 50 years, they do PSA testing in the annual physical thing. So usually what happens in US, that everybody has insurance, or everybody is supposed to have insurance, right? Because otherwise the healthcare is so expensive, nobody can afford it. If you go for blood, just simple blood test, it would be thousands of dollars, so it's just very expensive. So everyone has it. So insurance company realized that they are spending a lot of money on healthcare. So now the physical, annual physical thing, we had to pay some money for annual physical, just very little money, but now they pay you to get your annual. So you don't pay anything, but they pay you something. It's because a lot of diseases are caught at that stage. And then the person lives and the insurance company pays a lot more money. So all the companies which are giving you health insurance, they pay you money to get your annual physical. So in annual physical for men, after a certain age, PSA testing is also part of the annual thing. So in prostate cancer, one of the earliest thing is that PSA levels increase. So, and sometimes it is benign, it has nothing to do with cancer, it just increased, right? So when that happens, immediately the person gets tested. And if that person has cancer by any chance, they can remove it surgically. So if it is caught at an early stage, it's 100% removal, and the person can live for many years. In other cases, what happens, even if it is surgically removed and nothing is left, sometimes the cancer comes back, and that is very bad, and that is called castration-resistant prostate cancer. That is pretty aggressive cancer. And then, we means scientists developed very good drugs for them, like excellent drugs for them, because the signaling pathways are known. So there were only two kinds of cancer before, like non-castration resistant, the hormone sensitive one, and the castration resistant. Only two kinds of cancer existed. There was no third kind for prostate. So very good drugs were developed for castration resistant prostate cancers, and it completely shrinks, just completely goes away. But then what happened? That those patients who were exposed to those drugs now developed a third kind of cancer, which is called NEPC, neuroendocrine prostate cancer. So this cancer is man-made cancer. These cancer cells, they develop a new kind of resistance where they change their phenotype. Now they don't even look like prostate cancer cells, they look like brain cells, neuroendocrine cells. Signaling pathways are completely changed. So now research is not being on hormone sensitive or CRPC, they are going on neuroendocrine prostate cancer. What is going wrong there? So we know a lot about that too, and now therapy is being developed for that. So the problem is this, that we treat cancer and the cancer becomes even more different cancer. So basically, initially we were getting our food from this place, used to stop that supply, now they are doing something else, you know. So that's what it is. So, but keep in mind, we have made huge progress in cancer field. Initially somebody had cancer, that was a death sentence. Now vast majority of people live, vast majority of people live. So it's, cancer is not winning the case, they are not winning, we are winning. They are changing, but we are getting more and more clever also to know, yeah, you are going to change, but we are ready for you. Okay, so humanity is still winning. They are changing, more variety, but we are still ahead of cancer. Okay? All right. Now I come to my favorite topic, protein kinases. Okay? So this is regulating the regulators. Why? Because one kinase regulates 100 different proteins, which can be kinases or which can be something else. So now we are, it's like controlling the kings who are running the show. These are the kinases. So I'll tell you what they are. Meet the proteins responsible for more than 100 billion in cancer drug deals, just 100. So Gleevec targets a kinase, okay? So these are the biggest family of druggable targets, okay? And there's huge money in this thing. So what are kinases? Kinases are those proteins which use ATP. This is my personal figure that I use in my own talk. So in this one I have used some pictures for my own presentation. So what are kinases? And some of these are made by my students. So kinases are a superfamily of proteins, okay? Superfamily means there are many proteins in one family, and in human there are 538. In yeast, for example, there are 120 kinases, something like that. Okay, plants also have kinases. So these are a common family of proteins across the whole kingdom. And what they do, they use ATP and take the gamma phosphate of ATP and put it on substrates. So these are the substrates. Okay, so what that means, substrates. So I'm talking about protein kinases. So it is estimated humans have 100,000 proteins in their cell. Okay, and out of this 80,000 proteins are phosphorylated. So let's say this is a protein, right? So what happens when kinases put the phosphate here, this protein will go and anchor itself to the cell membrane. Or it puts the phosphate, it becomes active. Or it puts a phosphate, it will become inactive. Or it puts a phosphate, and because the phosphate is, now it will bind to other proteins. So basically the whole signaling is changed. So kinases are the main orchestrators of cell signaling. Is it making sense? So I'm talking to you and you are following what I'm saying. Kinases are doing their job because they are involved in learning and memory formation. Kinases are everywhere. I am doing my hand like this. Kinases are doing digestion, seeing, cardiovascular diseases, everywhere. Every cell has numerous kinases. I corrected myself from saying 538 because some kinases are not expressed in every single cell. But there are a total of 538 kinases in humans. And anything you can think of, they are involved. They are shown in black. And any disease you can think of, they are involved. Any disease you can think of, right? So the diseases are shown in red. So in cancer, obviously, they are a huge deal. So look at this thing. These are, on January 1st, 2022, more than 106 kinase targeting agents worldwide, including 72 US FDA-approved small molecule kinase inhibitors and 3 mTOR inhibitors were approved for clinical stage. Okay, so huge number. So in 2001, Gleevec was approved. Imatinib, Imatinib is Gleevec. These are antibodies there, but this was the first small molecule inhibitor, Imatinib here. And after that, just see how fast the approval is going. Okay, and this is just January 22. Okay, now there are a lot more. I attended a meeting sometime, I think in 2019, right before COVID, and they said 50, somebody said 47 or 48 kinase inhibitors have been FDA approved, and then Tony Hunter, who is like the grandfather of kinase signaling, he said no, 50. Now they are over 80 as of today. Okay, and here it was 72, so very, very rapidly. So it's really, look at that. In 24, it was 62.75 billion, and it's just going up and up. This is estimated. CAGR, I'm not a finance person, so I was thinking, what is CAGR? So I wrote it down, compound annual growth rate. I checked it once, and then I again forgot. Then I checked it again, then I again forgot. Then I finally wrote it here. Okay, this is what it is. So basically, it is just very rapidly increasing. Okay. There are 600, more than 600 active kinase targeting trials, focuses on combination, biomarker driven design. If this kinase is overexpressed, you give that drug to that patient. Okay, so this is kinase signaling, substrate protein 538 kinases in humans, I'm just talking about humans because we are talking about disease in human, but it is expressed everywhere. Fish, any organism you can think of, fly, mosquito, C. elegans, fish, amphibians, reptiles, birds, everywhere, plants, everyone has kinases. Okay, we estimate more than 80K proteins are phosphorylated by kinases, and also one substrate, if this is a substrate, if this is a protein, kinases can phosphorylate here, it will have a different effect, here different effect, here different effect, combination different effect. So it's very complex story here. Okay, so look at this thing, kinases, you see kinases everywhere. Without going into details, since we are focusing on cancer, kinases are also very important in neurodegenerative diseases, every single thing, cardiovascular diseases. But coming back to cancer, kinases affect cell survival, cell motility. Motility means metastasis, more metastasis. Evading immunity, kinases are doing proliferation, kinases are doing DNA damage response, kinases are doing, okay, angiogenesis, kinases, metabolism, kinases. Okay, this is a picture from my own review article. I actually made all these pictures, and this is one of the figure from that review article. This is on prostate cancer. So I'm not going to tell you, these are all the this is the pathway we discovered in our lab. This one, this one, this one, these are all our lab's work. But the point I'm trying to make here is that, see, this is aurora kinase. So we feel very close to kinases, so we make like more with eyes and everything. So you can see the eyes here, okay. So this is just aurora review article, but the point I'm trying to make here is that don't go in the details, but they are master regulators. They are, you see how many proteins are regulated by kinases just in one figure, just with one kinase. So that's what it is. If you control them, you are actually controlling numerous signaling pathways at once. That's the point of this particular slide. Okay, so that is what it is saying, regulating the regulators here. Okay. So why kinases are ideal precision drug targets? Keep in mind, we stop at 10:30. Whenever Leknath, keep an eye, 10:30, tell us, okay? So they are central regulators of growth, metabolism, differentiation, cell death, everything. And it is very common. So, kinases are very tightly regulated in the cell because they are so important. But kinases are often deregulated, and that is how they give rise to disease. The good guys become the bad guys, and that's what. So they are ideal drug targets, so precision therapy, okay. BCR-ABL, we know, it is successfully targeted with imatinib. Large family, large selection of kinases. The ATP pocket is extremely druggable. You can make inhibitors. Leknath has worked on it, right, in our lab and found some really good inhibitors for kinases. Excellent druggability, proven access. Now see, more than 80 plus FDA approved drug. And there are hundreds of drugs in clinical trial. And there are multiple inhibition mode. So do you guys know what is an allosteric inhibitor? All of you? There is anyone who doesn't know? Leknath, do you know what is allosteric?
Last time you were explaining, so
You remember that, right? Okay, good. All of you know it. So now, you don't know that? Yeah, don't hesitate to ask, okay? So, for example, kinases have this pocket. This is the enzyme site. So, whenever you are targeting the enzyme site, that is, catalytic site, that is competitive inhibitor, because substrate binds here and your inhibitor has to compete for the substrate. So, those are called competitive inhibitors. But allosteric means it is binding to some other site, and today I will show you some beautiful pictures of those. So, it is binding here, but the pocket is closing here. That is allostery. So it's not binding here, it's not competing there, but it is closing the pocket. Sometimes what happens, it could be here, 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 And very recently they have found that inside the pocket there is a small pocket, and if you target that small pocket, substrate is not going there but the inhibitor can go there, that also can close it. So but that is also allostery. Okay, so for the longest time people were just doing the ATP active site. So the problem was that 538 kinases all bind ATP. So although they are slightly different but not very different, right? So specificity was becoming a problem. Now we have seen, and that is also was first discovered by my postdoc advisor, that are these allosteric sites in the kinases. Okay, and if you target that, then you can make it even more specific for a given kinase. Okay. Today his name will come again in the next lecture because in G proteins 1. Okay. So that is why kinases are top precision drug targets. First of all, they are extremely important. Second thing, like BCR, able, Imanitab, we have 80 plus FDA approved. This is very wrong spelling. You can say this is made by AI. Okay. And favorable druggability and versatility. So these are extremely, so cancer targeting has transformed cancer in immune disease therapy. Okay. So now, why? One of the hallmarks of precision therapy is that you do the biomarker test. If a patient comes, you take a biopsy and you find out what's wrong with that patient. And based on that biomarker, you treat it, right? So if a patient comes and you see BCR-ABL is wrong, you give Gleevec. If a patient comes and Aurora A is, remember I told you about Oncotype DX testing in biomarker? That tests Aurora A kinase, and if Aurora is wrong, you give Aurora A drug. You do, so basically you are looking at CDK2 is one kinase, which is often, it's a cell cycle kinase, and it is a hyperactive in cancer cells. So if it is CDK2, you give CDK2 drug. So kinases are really biomarker driven because it's not a generic thing, it is specific thing, right? And if you know which kinase is wrong, you give the drug for that kinase. Makes sense? So that's what it is saying. It is the molecular biomarker thing. It is not, this is prostate cancer, give this thing. 100 different things can be wrong in prostate cancer, or 10 different kinases could be wrong in prostate cancer. So you don't give all the 10, you find out which one is the guy wrong, which guy is wrong, and then you give that drug. Okay? So you do genomic, proteomic alteration, EGFR. You know it is a kinase. EGFR is a kinase. BRAF is a kinase. ALK is a kinase. HER2 is a kinase. RAS is not a kinase. Tomorrow we'll start with KRAS. It's a very interesting story. Okay? So you use these things to guide therapy, enables targeted therapies that attack tumor-specific vulnerabilities. What happens is that, suppose there is a kinase which overexpresses Aurora A, right? So that kinase will be addicted to Aurora A. Okay, so suppose in any institute or any organization or in any kind of country also, if one person becomes all more powerful, like the whole thing is dependent on that. So if you attack that one most powerful person, if the whole thing is driven by that, you basically break the whole chain of command, right? So it's the same thing. Kinases become too addicted to some oncogenes, and if you can figure that oncogene out, right, then you can basically disassemble the whole thing. Okay, before they figure out now this person is gone, now another person can take over. Before the other person can take over, you have to kill it. That is what is resistance. If you give them enough time, you will develop resistance. Okay. See why our body is like that. That's how we survive. If something goes wrong, the body will do something, right? Body, these are living cells, so they are engineered this way to live, right? But then cancer cells use it for their own good. So that enables targeted therapy that attack tumor-specific vulnerabilities, improves response rate, reduces toxicity, because you are not giving chemotherapy, you are giving one drug against one kinase. Okay, so it extends survival, real-time adaptation through molecular profiling. So suppose you are giving Gleevec, right? So what will you do for Gleevec? How will you know if Gleevec is working? Tell me. How will you know Gleevec is working? You will check the expression of BCR-ABL through real-time PCR, because you know when you click Gleevec, it goes down. So you know, and that would be pharmacodynamic biomarker. Very good, guys. Okay, so drives development of kinase inhibitors, antibody drug conjugate and combination therapy. So they are the ones which are fulfilling this precision therapy thing, right drug, right patient, right time. Okay, so I told you in humans there are 538 kinases, and they are classified. There are many ways to classify the kinases, but the easiest classification is what they do, right? So based on that, they are classified into 3 groups: serine threonine kinases, tyrosine kinases, and dual specificity kinases. So tyrosine kinases, you know there are 20 amino acids, right? So tyrosine kinases are those kinases which phosphorylate on a tyrosine residue, and you know tyrosine structure, right? CH2 and then the phenyl ring and OH group, right? So this is the tyrosine group, OH group at the para position. So the phosphate group is added here. This is the peptide chain. So these are tyrosine kinases which phosphorylate at tyrosine, and then there are kinases which phosphorylate at serine residue. This is the serine side chain, or threonine. Okay, so serine, threonine, and tyrosine. So those kinases which phosphorylate at serine can also phosphorylate at threonine because they are very similar in structure. So they are called serine threonine kinases. These are the biggest family of kinases. Then there are tyrosine kinases. Usually when they phosphorylate tyrosine, they don't phosphorylate serine threonine. However, there are dual specificity kinases which can phosphorylate serine threonine as well as tyrosine. Okay. So tyrosine kinases, they are further divided into receptor associated tyrosine kinases and non-receptor. So receptor means something which is at the cell surface. So there are many different kinds of tyrosine kinase receptors which are cell surface receptors, and there are numerous receptors. Where is the cover? Yeah. So there are numerous kinases which are cytosolic or nuclear tyrosine kinases. So don't worry about the classification, but just to focus on this one, receptor tyrosine kinases. See, different kinds of receptor tyrosine kinases. So the important part is that this is the cell membrane, right? Look at that, they have different kinds of receptors, okay? Different kinds of receptors, but all of them, they have this membrane anchoring part here, this one here, and then this is called juxta-membrane region. All of them have juxta-membrane region, and this part which says TK, this is the tyrosine kinase domain, okay? So see, all of them have tyrosine kinase domain. Some of them have 2, okay, some have 1, some have another structure after the tyrosine kinases. So oftentimes, not all the time, oftentimes what happens that these tyrosine kinase domains are inactive till the ligand binds on the top, and when the ligand binds on the top, these kinases, they become active, okay? So see, these are TKs are tyrosine kinases and all different kinds of domains, and this is showing you all different kinds of domains here, okay, here. So look at this one here. So why I am specifically focusing on receptor, RTK means receptor tyrosine kinase. Why I am focusing on receptor tyrosine kinases? Because many of these receptor tyrosine kinases are deregulated in cancer. EGFR, HER2, they are all those kinds of things. So usually what happens that these tyrosine kinase receptors, they exist as monomer, separate, no activity, switched off, okay? Then the ligand comes and bind and they dimerize, and when they dimerize, these two domains, they come in close contact with each other. So one phosphorylates the other and the other phosphorylates the first one. So this one will phosphorylate this guy will phosphorylate this one. They usually cannot autophosphorylate themselves, so this will phosphorylate this will phosphorylate this, and then they will become active. And when they add the phosphate groups, P stands for phosphate, they are like glues, molecular glues. Then other proteins will come and bind here, and the signaling will turn on. So let's say here in this room, right, you guys are all gathered. So because you are all gathered, we are all talking to each other, right, crosstalk. So in signaling, what happens, unless you get different kinds of proteins in one platform, they are not going to talk to each other. So after this phosphorylation, these different proteins come and dock at specific sites. Now this protein can talk to this protein can talk to this protein, and this protein can talk to this protein. So this is a very simplified version, but usually what happens, one tyrosine kinase domain can bind 10-15 different proteins. This guy also 10-15 different proteins, and these 10 to 15 proteins are all talking to each other, right? So it provides a platform for everyone to gather together, and that's how signal is turned on. So what goes wrong in cancer as far as these RTKs are concerned, receptor tyrosine kinases? So see, they are overexpressed. If they are overexpressed, their kinase activity, suppose there are two monomers here. If they are not overexpressed, they are far apart. But if they are overexpressed, they are forced to come together like this. And then in the absence of ligand also, they will activate themselves because they are in close proximity. Okay, see here, sometimes what happens, there are some mutations in the receptor tyrosine kinase that they don't even need ligand binding, they are just active all the time. So this is gain of function mutation. Gain of function means increase in activity, gain in activity. Okay, this is normal. Now here chromosomal translocation fusion protein, some kind of translocation has occurred that has activated them permanently. Autocrine activity, now this is very smart, prostate cancer cells do this thing. They make their own ligand. Ligand is supposed to come from outside, not from themselves, but they make their own ligand, keep activating themselves. This is also very common. So all of these things will lead to hyperactivation of RTK, which will lead to cancer. Okay, so these are another mechanisms. So see, gain of function mutation. This guy is monomer, but it is still active. It doesn't even need the second partner. Here, amplification, they are just expressed in huge amount. Some kind of rearrangement, they are always active. Okay, so see this one here, initially they were standing like this, waiting for the growth factor to come and activate them. Now they don't even need the growth factor. They go inside, they are always active. See, this guy doesn't even need the growth factor. So this is they are down here, they are active. Okay, so see, blue one is the tyrosine kinase domain, and these are the phosphates which are added. So under normal circumstances, phosphate will only add when the ligand binds, but this monomer has the phosphate already, it's already active. These guys are over amplified, hugely expressed. Here, it doesn't even need this whole thing. This is active, this will go inside. Here, one domain became two domain. Duplication, see this is a monomer, but it has two tyrosine kinase domain. All of these have one each, right? They are dimerized, but they have one each. This guy has two of them. Here they are making their own ligand and activating themselves. Okay, so because of that, they are deregulated. Okay, so that was receptor tyrosine kinases. But other than that, there are cytosolic kinases also. They are also activated on their own. So this is BCR-ABL, which is a cytosolic kinase. You guys know chromosomal translocation in BCR-ABL, right? So they are like ABL, ALK, FGR, PDGFR. They have chromosomal translocation. They become hyperactive. ERB is HER2 receptor, overexpressed. KD stands for kinase domain. So all of them have kinase domain. We are talking about kinases here. Okay, not necessarily tyrosine kinases, all kinases. Overexpressed, so now they are activating. Point mutation, this one overexpression here, B. So look at this overexpression. Then there is some point mutation, because of that they are activating themselves or they are activating others. This is a very interesting part. So remember I said kinases are very important, so they have to be tightly regulated. So one of the way, this is a very famous kinase, Src kinase. So this is the kinase domain. These are other protein-protein interaction domain. So Src has a tail where there is a tyrosine residue, and this is tyrosine 527, which is phosphorylated. So once it is phosphorylated, it binds its own SH2 domain, and kinase domain switches off like this. Okay. So because of the tail thing, so if I have to draw this structure here, so this is catalytic domain, okay, of Src. So this is the catalytic domain of Src, and this is the tail here, and this tail has a tyrosine residue which is phosphorylated, okay. And then there is this other domain called SH2, and then there is another domain called SH3. Sorry, I have to draw it in slightly different manner. I am drawing it exactly the way the crystal structure has shown Src binds. So then this thing comes like a tail here, and here is SH2 domain, and then here is SH3 domain, okay. So this is the catalytic domain. So SH2 domain binds the tail, the tyrosine phosphorylated, and when it binds the tail, this is switched off like this. Do you see that? So if you look at this picture, when SH2 domain binds the tail, this is switched off, okay, and the kinase is inactive. So what cancer cells do? They remove the tail here. Now the whole thing is open like this, and it is always active. Okay, so basically the point is that they mutate themselves to keep themselves active all the time. Okay, so now let's say, how do we target kinases? So this part will introduce you to a lot of different things, including PROTACs. Yes, Evans.
Is there characteristics of the tyrosine protein? Okay, when protein and protein is interacting, two proteins are interacting through phosphorylation. Is there a characteristic?
Good, thank you. Yeah, repeat your question again.
Okay, when two proteins are interacting through phosphorylated protein, so that tyrosine that is phosphorylated, is there characteristics
very
of
good
the sequence,
question.
like
Very good question.
amino acids?
Yeah, this is very good question. So what happens is that let me go back to the previous one here. So when I was a postdoc, I worked on this protein, Src kinase. So I can even tell you the sequence which binds there. I remember the sequence because somebody in our lab made the peptide with that sequence. So what happens is that when a tyrosine residue is phosphorylated, it binds SH2 domain. So SH2 domain, see the SH2 domain shown in yellow. So that this thing, SH2 domain, will not bind tyrosine unless it is phosphorylated. But then there are different kinds of SH2 domains which have different sequence specificity. So only tyrosine phosphorylation is not enough for SH2 domain to bind. It will also need a sequence here and here. So for Src, that sequence is E, E, I, isoleucine, Y is tyrosine, G, E, F. The sequence for Src, this thing is, so they don't care about this sequence, E, I, Y, G, E, F. And this is the tyrosine which is phosphorylated. Okay. So Src SH2 domain, so see this protein is Src here. Okay. So Src SH2 domain will bind tyrosine here if it has this sequence here. Okay. And if you make small modification, it will still bind. So if you change isoleucine with some other hydrophobic amino acid, it will bind but not as tightly. Okay, so different SH2 domain will bind phosphorylated tyrosine, but with different specificity around it. But they usually at, so if you have to read this sequence, This is the site which is it is binding. So this is +1 site, +2 site, +3 site. This is -1 site, -2 site, right? This is how we name it. So at -1 site, it has to have a large hydrophobic amino acid. If you make glycine, it will not bind. So although it loves tyrosine, phospholylated tyrosine, but it has to have specific sequences to bind. So does that answer your question? So SH2 domains are involved in protein-protein interaction, and they will bind phosphotyrosine residues with specific sequences around them, just proximal to the site, not very far, just right then and there. Okay? Okay, so let's talk about how do we target kinases, and that's where the thing I have been telling you about PROTACs will come into picture, and nanobodies, which you have not heard, and these are super exciting things. Okay, so small molecular inhibitors, there are thousands and thousands and thousands of protein kinase inhibitors. 80 plus are already FDA approved, right? So we have many small molecular inhibitors for kinase, Gleevec for BCR-ABL, right? You guys know that. So this is, we have huge amount of small molecular inhibitors. So these are low molecular weight drugs, small size, so they can even go blood-brain barrier, no problem. So this is very advantageous. They can go deep inside the tumor. Okay, they are extremely easy to target. Okay, target specific. They are designed to bind to the active sites or even at the allosteric sites. They are often reversible because these are small molecular inhibitors. Unless you make them covalent, they will go there. They can also come off, but you don't want them to come off, so you make them very, very powerful, very, very tight binders. And they are usually orally available. You don't have to get IV. Gleevec, just pop the pill. So they are very useful, very easy to handle. And they are also cheaper in that because you don't have to go to the hospital to get it administered. So they typically inhibit signaling pathways by inhibiting the enzymatic activity. So for if it is EGFR, which is a receptor tyrosine kinase, you just give it to them, it will inhibit the activity of that enzyme. Okay. Tyrosine kinase inhibitors like BCR-ABL, imatinib, and all of those things. So small molecular inhibitors are tiny drugs that enter cells and shut down disease-driving things. So that's very easy. Now this is something interesting. When I was telling you about allosteric and Lekna, this will make much sense to you as a chemist. So kinases, they exist in active form and inactive form. Okay. So there is this loop kind of structure in kinases which has this sequence DFG. Okay. So if DFG in and DFG out. So this is the active form of kinase, this is the inactive form of kinase. So if the loop is inside, the kinase is active. Active means it can phosphorylate proteins. If the loop is out, kinase is inactive, it cannot do any job. So there are many different kinds of kinase inhibitors. So there are inhibitors which are called type 1 inhibitors, which will target the active form of kinase. Then there are, so keep in mind, anytime there is DFG in and DFG out, the inside pocket shape is different. So DFG in is this, DFG out is this. I'm just making up my hand, it is not exactly this shape, but the point I'm trying to make is that these are different shapes of pocket. Okay. So some inhibitors will target active sites, some inhibitors will target the inactive form of kinases. So type 1 will inhibit the active form, type 2, this is type 1, this is type 2, so active, inactive form. Then see, there is an allosteric pocket within the ATP binding pocket, right? So as I was telling you, allosteric can be here, but can be also inside. So type 3 inhibitors will bind a deep pocket inside the active site. So ATP will bind here, it does not have access to that, but this is an allosteric, this is type 3. Now this is also an allosteric site, but far away from here. Okay. So it will bind here, but not at this pocket, but a pocket far away. But this pocket is changing the shape of this one here. So see here, this is a different allosteric site right here. Now type 5, both of them are binding and they are somehow linked together. And type 6 is that they are both binding, but they are not linked together. So you have 6 ways. I have seen this field evolve in the last 10 years. 10 years ago, nothing was known like this. Okay, so this is another picture. Type DFG in active kinase, it is binding here. Type 2 DFG out, it is binding here. Type 3 is the allosteric pocket within the pocket. Okay, type 4 allosteric site far away from the active site. Here type 5 bivalent, type 6 this one. Can you see that, all of you? So makes sense? Okay, so this is just, so what do kinases bind? ATP. ATP is purine ring. So when people make inhibitors, they make purine based compounds. So see, most of them are purine, at least you know they are bicyclic compounds. This is purine, but then they have made mimics of that. So these are different scaffolds which are extremely popular in kinase field. Okay, diverse thing. Many of them are like this, 14% quinazolone thing. You don't have to remember anything, but just to tell you the scaffolds. So you would imagine these are mostly competitive inhibitors because they are resembling the purine ring of ATP. Okay, so look at the approval rate here, so rapidly. Okay, this is Gleevec in 2001, first one, and then after that so many have been approved, over 80 different kinase inhibitors. This is yet another one. See how many are approved. So it is exponentially increasing every year, okay, and huge amount of money in this thing. Initially, look at that, initially most of them were ATP binding site competitive inhibitor, pink, 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 and now ATP binding site non-competitive inhibitor. They are targeting the allosteric sites, okay. Now, this is if you go through a pathway. Tomorrow we'll really focus on this GTP RAS. This is yet another class of superfamily of proteins. Okay, but this side you can see kinase inhibitors. This is a kinase, this is a kinase, this is a kinase, this is a kinase. And look at that, how many inhibitors are there, okay, against kinases. So these are the famous inhibitors, these are the famous receptor tyrosine kinases which are highly deregulated in cancer. So this is, so usually what happens if you give a drug, the receptor mutates or the receptor is already mutated in cancer. So this is a famous mutation, threonine 790 is converted to methionine, or this L848 is converted to arginine. So these are specific inhibitors. This is specific for this mutation, this is specific for this mutation. This is tucatinib. These guys, look at this. These are all small molecule inhibitors. PI3K is also a kind of kinase, but different from protein kinase family because it's a lipid kinase. It has its own inhibitors. This is a kinase inhibitor. See how many kinase inhibitors are there. Okay, this is the famous kinome tree. So all the kinases are divided into different families, and this is the kinome tree. Can you see that here? Okay. So these are, you know, different phylogenetic trees. You know how the kinases are classified based on their sequences. So at all of these things, look at this, the number of drugs. It's this huge number of drugs against the kinomes. Okay, this is just different companies are developing, they have different drugs in different phases of clinical trial. Okay, now we come to PROTAC. I'm sure you guys are waiting anxiously for PROTAC. So before I tell you about PROTAC, tell me how many of you know about the ubiquitination machinery? Anyone knows about ubiquitination machinery? Raise your hand if anyone knows about ubiquitination machinery. Only you. So two of us will make everyone else understand this machinery. Okay? All right. So this is very exciting, okay? So I want you guys to think about it. If protein A is bad, let's say one kinase is bad, okay? So what will you do to inhibit that kinase? What will you do to target that kinase? You will say that, don't worry about it. If one guy is bad, you inhibit that kinase, right? But let's say this guy is a kinase and this is bad, and this is the ATP binding site here. So you inhibit that, but this kinase is also bad because it is, remember, kinases can also act as platforms. So you have inhibited its activity, so it cannot phosphorylate, but this can still act as a platform to assemble all the bad guys together, right? So even if you inhibit it, it's not going to be very effective. Do you guys address? So what will you do in that case? Degrade it completely. And PROTACs are the outstanding way of degrading a protein completely. You make them disappear at the protein stage. See guys, understand something. There is a DNA, then there is RNA, and then there is a protein. So if you don't want some protein to come, you can degrade the RNA, and that would come under RNAi or CRISPR or something there. CRISPR works at the DNA stage, RNAi works at the RNA stage. CRISPR is good for the DNA. You just delete the gene at the at the DNA stage. That is CRISPR. If the RNA is there, you can degrade it by siRNA. But if the protein is there, you can degrade it by PROTAC stage. So this PROTAC was developed by Craig Crews. He's at Yale University, and it was developed in the early 2000s. And the moment it was developed, it just rose like extremely steep. And now there are so many PROTACs in clinical trial. I think the first PROTAC will be FDA approved in 2026, next year. But they are in late stage clinical trials, and next 2-3 years, many PROTACs will be FDA approved. Okay, so these are very powerful way. But to understand PROTACs, we have to first understand the cellular ubiquitination machinery. So what is that? So suppose you are looking at the protein levels of any protein in the cell, any protein, it has some certain level, right? Whatever level it is, high, low, whatever it is. So this level is maintained by two things, synthesis and degradation. So far you guys have understood everything which is, when things become defective, they have to die. You don't want to have defective things hanging around. Defective cell, they need to die. Defective protein, it needs to die. So all proteins have a half-life. Some proteins have a very short half-life, other proteins have a very long half-life. So the proteins need to degrade, and this degradation machinery is called ubiquitination machinery. Okay, so before I explain to you this slide, I will explain to you what exactly is ubiquitination there. So ubiquitination is a mechanism by which you degrade a protein, and it has three players which come into play: E1 enzyme, E2 enzyme, and E3 enzyme. Okay, so every protein has to be degraded eventually. So there are 2 E1 enzymes in humans, about 38 E2 enzymes, and then there are over 600 E3 enzymes. Okay, I'm going to skip this slide and come back to this in just a minute. So what happens is that there is a protein called ubiquitin which is shown here. So this has 76 amino acid. So these, what happens is that, remember, E1 enzyme will come and react with this ubiquitin protein using ATP. So this is the reaction. E1 has a SH group. I want you guys to understand this thing. So E1 has a SH group, thiol group. So this has a carboxylic group at the end. So carboxylic group, when carboxylic group reacts with alcohol, what is the bond which is formed? Ester bond. And if a thiol groups react with carboxylic group, what is the bond which is formed? Now the chemists have to step up. Thioester bond, right? Thioester bond is formed. So this is a thiol group, and thiol group is present in which amino acid? Cysteine. Very good, right? So obviously it's a cysteine residue, right? So cysteine residue reacts with carboxylic group of ubiquitin, just the last carboxylic group of ubiquitin. In the presence of ATP, AMP and pyrophosphate are released, and it forms thioester bond with ubiquitin, right? It is a generic reaction. Then another enzyme will come, E2, and this E2 will come, and this thio ester ubiquitin group will be transferred to E2, and now this is the one which is holding the ubiquitin with it, and E1 leaves there. Okay. Now what happens? E3 enzyme comes into play. Okay. E3 is very special because on one side it will bind E2 and on one side it will bind substrate. So keep in mind there are many substrates. So it is the E3 which will recognize the substrate and E2 on the one side. So it is providing a platform there, on one side recognizing the substrate and on the other side recognizing E2, and then it will transfer the ubiquitin here. Make sense? And once the ubiquitin is transferred, many more ubiquitins are transferred, so it will form a chain. And that is the kiss of death. This is called, when the Nobel laureates were given the Nobel Prize for this work, they said, okay, explain it to the rest of the world in very simple language. So they said basically what happens, there is this protein which acts as a kiss of death, and anytime that protein is added, that particular protein is condemned to death. Okay, that's what it means. So now this protein will be degraded by the proteasome. Proteasome will not recognize the protein, it will recognize this, and it will know it has to degrade it. So it will chop off this part so that this is recycled, but it will eat up the substrate and release all the amino acids. Okay, that is what it is. Do you guys understand this thing completely? Ask me if it is not clear. I think you have a question. Go ahead.
I'm confused at that step. Like in step 1, that E1 is taking ubiquitin. Then why we need E2?
So here is the thing. So suppose Professor Venkatesh is E3, okay, and there is another Venkatesh, he is another E3, okay, and Evans is another E3. So there are 600 E3s, okay. So Professor Venkatesh has access. Professor Venkatesh will recognize 100 different students. Younger Venkatesh will recognize another 100 students. Evans will have his own 100 students, right? So I am E1 and you are E2. So my job is that I will just take ubiquitin and I will attach it to myself. I don't have access to these guys because these guys don't recognize me, but they recognize you. Okay, you are from IIT Indore. So I give my thioester ubiquitin to you and it is there. I don't have access to these people, but you do. So you go to Professor Venkatesh, you also go to Venkatesh, and you also go to Evans. Okay, my job is just to give it to you. I am out of the picture. That's why there is only 2 of me. There are only 2 E1. There are a lot more of you because you guys are talking. Now actually I cited 3 of them, but there are 600 of them. So what is the difference between these 600? They have different substrate specificity. The 100 here, the 100 here, the 100 here, and the other 600, they all have their own hundreds there. I have nothing to do with those 100. Now what you have to do, you cannot interact with those hundreds either. So Professor Venkatesh on one hand will recognize you and the other 100 proteins. Venkatesh will recognize you and the other hundreds. Evans will recognize you and the other hundreds. You see, so my job is most generic. I just have to take ubiquitin, just throw it to you. So I have nothing to else. So I don't have to have 10 different ways because I'm not interacting with anyone. But E3, these guys are interacting with 100 ones. So there are many E3s because they have to have their own group of people. What is common between you and them? Because they all recognize you. So they all have some place for you to bind. Do you see that? They don't have space for me to bind. So I cannot go and bind there, but they recognize you. All the 600 people, they recognize you. Okay, so now let's come to that. There is only 2 of me, and I am just taking ubiquitin and transferring it to. There are 38 of E2, so there are 38 people there. So my job, there are only 2 of us, 2 of me. I'm transferring it to 38 of this thing. Okay, these 38 of these things now react with E3. There are over 600 of them. Okay, so my job is done as soon as I transfer it to you. I go and I keep on doing that. So my job is like I'm just transferring it to E2s. That E2 will go to E3, and E3 has, see, one place for E2 to bind, one place for the substrate to bind. And each E3 will have their own sets of substrates, maybe hundreds of them. So they are designed in such a way that they will recognize only their own 100 proteins. What they are doing? They are bringing these guys together. E3 does not have the capability to itself get ubiquitinated, but by binding E2, now this will be transferred. Do you see that point here? All of you? So this is how it works. So now if I go back to this slide, it makes sense. Less of E1 enzyme because they have a generic function. More of E2 and a lot more of E3 enzymes. Do you see? Because E3 are the one which will recognize every protein in the cell. And each E3 will have their own sets of proteins to be recognized. So that is why you have, so that is where the substrate specificity part comes in. And there are three major kinds of E3 proteins. Let's not worry about it. They are ring kind of, these are called ubiquitin ligase, ring, HECT, and RBR set of proteins. And this is ubiquitin itself, which is shown as UB. Do you guys follow this so far? You have to understand this thing before you understand PROTAC. And then what happens, I don't have it here. I should have the proteasome picture also, but basically this will be picked up by the proteasome and degraded. And proteasome is like a barrel. So on one end the protein will end, and from down all the amino acids will come out and they are recycled again. Okay, makes sense? So now let's go back to this slide here. So what this smart person did, so suppose you have BCR-ABL on one side. Which small molecule will bind BCR-ABL? Tell me. Gleevec, right? So you take Gleevec on one side. So what will happen? And then on Gleevec on one side, and it will bind BCR-ABL. Then you make a small linker just to give it some room. You don't want steric hindrance, right? So you give a linker. On other side, you directly bring a E3 ligase there, right? So E3 ligase is once again this guy here, this E3. So you bind the substrate on one side. No, you bring the E3 ligase here. So this one end will bind the protein of interest. So if it is BCR-ABL, this is green part is Gleevec, and this part is a ligand for E3 ligase to bring E3 ligase here. Okay, so if you bring the E3 ligase, E2 is already attached to E3 ligase. So this protein is in close proximity, it will ubiquitinate it, the proteasome will degrade it. Is it making sense to all of you? Anyone who doesn't understand, I'm very happy to repeat it. Just raise your hand. Okay, please do it without my prompting. If you don't understand, ask me what is the thing you don't understand. I'll explain again. So suppose this is E3.
I did understand that, the Gleevec gets attached to it, and then on the other hand there's like another ligand. Like, what is the second step to it? That is something I didn't understand.
Let me say that here. So this is E3 ligase here, you understand there. So you understand there. So what you think is that, now think about it, you are trying to say where are you going to hook something to bring E3 there. So imagine that E3 also has a pocket here. So what you are doing, you are making a bifunctional molecule. One is targeting E3 directly, not here, not here. Okay. The substrate, see, the problem is this, if you activate E3 randomly, it will degrade everything, right? So you don't want that. You want to degrade one protein of interest, any one protein of interest. So you have to trick that protein to come near E3. If it comes near E3, E3 will degrade. Okay. So what you do is that all E3s have their own pocket, which is not shown here. So suppose this pocket exists here, and that pocket also binds some ligand, or you can make a small molecule that binds the ligand, that binds that E3. Then what you do, you take that part and you take a linker and you attach Gleevec, for example. So Gleevec will bind BCR-ABL, and that ligand will bind here. So that BCR-able is in close proximity of EDIT, it will switch it here, it will be ubiquitinated, it will be degraded. Is it making sense?
Yeah.
All of you? All of you? So what I did not say, and that is why you got confused, all the E3 have their own pockets also, which is not shown in this slide. So you are using the ligand for that pocket to bring the two things together. It's like you take a person in front of the Jallad, and the person will kill that person. Okay, that's the point here. If you take it to the place where death is occurring, the guy will get degraded. Okay, so so here, let's say here, so it has three things: protein of interest ligand, E3 ligase ligand, and you always make a linker. As an organic chemist, we know if you make a linker, there is a flexibility. If you make them next to each other, they may not come close to each other because of steric hindrance. So you have to have a linker in between. So this is protein of interest linker and E3 ligase. So now this complex forms, and now because E3 ligase is always attached to E2, remember? E3 ligase is always attached to E2 here. So because of that, now the ubiquitin will be transferred, and then this will be tagged to the protein, and then this is killed. Isn't that very simple? It's a very, very powerful way. Okay. So this is, so now there is, so go ahead, Evans.
So what is the order? Because these are two proteins.
Yeah.
And the compound is targeting two proteins. So what is the order? Does it bind to E3 first, or it binds to the protein of interest? What is the order?
It can be either way, but you have asked a very good question, and I was not going to say that in details because in interest of time. So whenever you have to do two proteins, if you bring two proteins together, right, and if you add too much of the compound, I want you all guys to think about it. So let's say you have 100 molecules of BCR-able there, okay, and you add 500 molecules of this bifunctional ligand, what will happen? That's a very important question. So you have a bifunctional compound. On one end it will bind BCR-able, for example, other it will bind the E3 ligase. But if you add 500 molecules of the bifunctional ligand, when you have only 10 BCR-able and 10, let's say, you have only 100 BCR-able molecules and you have only 100 E3 ligase, and you have added 500 molecules of your bifunctional compound, then what will happen?
Do we have ligand like Gleevec binded to the tyrosine?
Gleevec binded to the tyrosine kinase, BCR-able, and
Then, because to attach to this bifunctional ligand, we need some ligand which will bring BCR-able to this bifunctional ligand. So those BCR-able which are already having ligand attached to them will come in proximity.
Let me correct you. If you have a because your question has high significance, so instead of answering it directly, I want you to think in other way. So let's say these two are the protein targets, right? So if I have huge amount of ligand, so one end will bind this, one end will bind this. So if you have huge amount of ligand, huge excess of ligand, it will bind, many ligands will bind this only, many ligands will bind this only. There is no reason for them to come in close contact again. So you have to use less ligand so that whoever binds this, order is not important to answer your question. The second part will come, because there is no excess ligand there. Is it making sense? Right? So that part is not that important, which one is binding first, because that will probably depend on the affinity, which is the higher affinity ligand. But the more important thing is, don't use huge excess of it. If you use huge excess of it, it is not going to function. So when you do this experiment, which we did in our lab, you have to do a titration to see which amount gives you the best degradation pattern. Because if you use too much, they are both bound, but they will never come in close contact with each other because it's huge amount. Right? Very good question. Okay, so before I go to PROTAC, this has nothing to do with PROTAC, but independent of PROTAC also, E3 ubiquitin ligases are considered as very important drug targets. Why? Because some ubiquitin ligases are the one which degrade all the tumor suppressors. And if you know these are degrading tumor suppressors, if you inhibit them, it would be good. But if they're degrading oncogenes, you don't want to inhibit them. Okay, so this is just saying that if they are degrading tumor suppressors, then they are targetable and then you want to inhibit it. Okay, or you can kill them using shRNA also. Okay, or you can kill them using PROTAC also. Okay, so here is another two kinds of different PROTACs. So this is a reversible inhibitor, so it binds to the active site. So this is a kinase BTK, so this binds here, but it binds reversibly, but it gets degraded. You can also make a covalent PROTAC. If you make a covalent PROTAC, sometimes you get better results because the binding is pretty tight there. So now you can understand PROTACs pretty easily. This is protein of interest. POI is protein of interest. This is the bifunctional molecule. So on one end it will bind the protein of interest, on the other hand it will bind the E3 ligase, and now ubiquitin will occur, proteasome will chop it up, this will be recycled. Isn't it a very neat method of degrading? Yes?
Will that degraded part will be taken by APC and then represented again?
No, this is not that part. These are amino acids. These are not even antigens.
Okay.
Antigens means some part of the protein or polypeptide which has a certain structure.
Which needs to be represented.
Yeah, so this is internally. This is so ubiquitination is occurring in our body all the time, and if it doesn't occur, we will die because a lot of clogging. So proteins have a half-life. After that, they get degraded because they get damaged. If they are damaged, they need to be degraded. Okay, some proteins should be present there for a very short period of time. If they stick for a long time, it is very dangerous. So proteins have this encoded feature which determines their half-life. Okay, so and they need to be degraded at time. So this is called homeostasis. Something is synthesized, something is degraded. The way human life, or at least life on this earth, is designed in such a way that there is always constant renewal all the time. So somewhere I read when we celebrate our birthday once a year, basically most of the cells are changed by that time in our body. They are not the same cells which were there a year ago because of the renewal, right? So dying is good because new things are replacing them, and then damage is not accumulating, right? So these proteins, when they're chopped up, they're not chopped as antigen, they're chopped as amino acid. There is no recognition there. This happens inside the cell, every cell of our body, okay? And then these amino acids are recycled to make new proteins, okay? But that is a good question. This has nothing to do with antigen, okay? Those cells are different. These are not APC cells, okay? These are every cell of our body, okay? So this is, now look at this thing. So this is a very complicated slide, but if you look at this, see the red part here, okay, and the blue part here. So let me look at the red and the blue part here. Yeah, so the red part is the one which is recognizing. So look at the blue part here. These blue parts are similar, right? So they are binding the same E3 enzyme, CRBN kind of thing. This is a E3 ligase, CRBN. So blue part is identical because it is bringing CRBN here. This is the linker. This is the linker, long linker, and these ligands are specific. So this ligand red is specific for HDAC8. This ligand is for BCL2, targeting BCL2. This is MCL1. Can you see that? So on one side, it is targeting the protein of interest, which is the red part here. Then the linker part is there. Linker is usually linear because that will give you flexibility, right? It is not very complicated linear. Linear is very simple. They add some oxygens in between. They just don't make it CH2, 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 CH2 because chemists have to say, why don't you make CH2, 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 CH2 all the way? because that will make it hydrophobic, and it is all water. If you make it all hydrophobic, everything will collapse on it like that. So that's why you put some oxygens in between. Okay, and then this is the ligand for CRBN, E3 ligase. In this thing, see, in this one, this is VHL ligand. So there is a E3 ligase which is called VHL. So this blue ligand binds VHL protein, and then you have unique ligands here. And then see the linkers everywhere. Can you see that? Makes sense to all of you? 100%? Raise your hand if you don't understand. Please raise your hands. Is it making sense? Please ask. I can see from your face.
Yeah, there's this one thing, those figures that you have listed about, like A.
Yeah.
I don't understand those. So you know that E3 ligases that you've written,
Yeah.
and there are like, so what are those images that you've attached there?
Okay, so what it is, so don't go into too much details, but I'll tell you what it is. So these are all E3 ligases, that's why they're saying E3 there. But see, this has HECT domain, and when you have HECT domain, then you have to make the ligand for HECT. This is part of the E3. So when I say you have to make a ligand for E3, there are 600 different E3s there. So you have to make ligands for those kind of things, right? So here it is ring domain. So this one here it is ring domain, this ring domain. Here it is ring domain. Here it is RBX domain. Look at this one here. Usually there are 3 major kinds of it. Ring domain doesn't mean only ring domain, but it has some other structures also. See, here it is HECT, here there are 2 ring domains, here it is 1 ring domain, here it is ring. So even rings are different from each other. This is ring 1, ring 1, ring 1, ring 2, this is ring. Okay, so what is there is that you have to make the ligand against these guys, but these guys are also slightly, like all kinases, they bind ATP, but ATP pockets are different from each other. So these are different E3 ligases, so it depends on which E3 ligase you are using at this end here. So this is the ligand for E3 ligase. Some of them will be against the ring domain, some of them will be against the HEC domain, some of them will be against the RBR domain. So that's how they are different. And VHL and CRN, these are name of specific E3 ligases. Okay, so now this method is so popular. I mean, it's unbelievably popular. So we have an idea which ones are going to work the best. So you don't have to do all 600. There are few good ones, and most of them people use either CRBN or VHL. They are considered the best ones because they work very well. Make sense? All right.
Yeah. So that blue part is already endogenous E3 ligand.
Exactly.
You have to give patient the another linker.
No. So what happens is that, I'm glad you asked, inside the cell is just the protein of interest and the E3 ligase. There is no ligand inside the protein. You are giving, your job is to bring them together. So you use the ligand for your protein of interest as a bait to bring your protein of interest, and you use the ligand for the E3, which is not endogenous, again as a bait, and you attach them together so that they are forced to come in close contact with each other, and you degrade them. There is nothing endogenous about the compound. That is man-made. Man has recognized the ligands for these two guys. Gleevec is man-made. So are these ligands for VHL and this thing. You are just making a bifunctional molecule to bring the two molecules thing. So suppose Professor Venkatesh has organized the Gyan course, and because of the Gyan course I am here, and because of the Gyan course you are there, right? So he has brought us together. So, but the thing is that if there is no Gyan course, we would have never met, right? So the same thing is there. This bifunctional molecule is not endogenous. Neither of both of its components are not endogenous. This is man-made, attached together, but you add it to the cell, the two proteins are forced to come together. You are forcing them to come together, but one just happens to be death. So if you bring death next to a protein of interest, that protein will die. It is as simple as that. Inside the cell, you just have two proteins, the death protein and the protein of interest. And you can take any protein of interest and you can do this thing. What is the limitation there?
Size.
No, what is the limitation of PROTAC in your opinion? Can you make a ligand of any protein of interest?
Ligand of
Specificity could be a very big issue. Good point. And what else?
Ma'am, because of huge size, the membrane permeability.
That is a very big thing. So I have few slides just on that thing. So let me ask you this thing. In a way, it is good you will get the slides tonight, so you cannot prepare in advance. So I can ask you the question in advance. So tell me, is it a small molecule or is it a biologic? This is your test. Is it a biologic PROTAC or is it a small molecule?
We have an antibody in the bifunctional ligand, and we have
There is no antibody here.
that bifunctional.
I can see this is a very new thing for you, so I recommend when you guys get the presentation today, please read it, because PROTAC is extremely important to know. Okay, so there is no antibody here. So now tell me, raise your hand if you think it is a small molecule. Very good. And raise your hand if you think it is. See, if you don't raise hand in either of them, you are on the failing side. So you have to raise your hand. Raise your hand if you think it is a biologic. Okay? And raise your hand if you think it is a small molecule. Okay, 3 of them. At least you also added in the small molecule thing. It is not. No. So right now I won't give you the answer. You have to choose one side. Okay, and then I'll explain why it is, whatever it is, why it is that it is. Small molecule. So let me ask you again, small molecule. 1, 2, 3, 4, 5, 6. And then I won't ask the biologics, because rest all of you fall into the category of biologics. If you don't participate, you are biologics here. Okay? Small molecule. Small molecules, yes. Okay, why it is a small molecule? Because there is no biologic attached to it. Did we make any part in the with DNA, RNA inside the living organism? No. But ligand is man-made. Exactly, it is a small molecule. Leknath, you and you, Venkatesh, 3 of you were small molecule from the very beginning, because it is a small molecule. The reason why others were confused about this not being a small molecule, because technically it is not a small molecule, because it is just a big molecule. It comes under the small molecule category, but it is actually a relatively bigger size small molecule. That's the thing which confused you all, but you understand that, right? And this is a very important distinction, and I'll spend some time on that. But is PROTAC part clear to all of you?
Yes.
Okay, good. And I'm very happy to explain it again if you don't understand it, okay? All right, so this is just to tell you only about kinase targeted PROTACs in 2025. These are the kinase targeted lead candidates. Look at them, they are already in phase 3, phase 2 clinical trials, and these are the kinds of cancer, and this is the clinical value of this. These are the names. I have not included the PROTACs for other targets. They are even more advanced than kinases. But by 26, 27, many of these will be approved. Okay. So targeting kinases via small molecule inhibitors and PROTACs. So these are just listed there, but I'm going to show you this part here. They are generally classified as small molecule, not biologics, because they are made of 2 small molecule ligands, one for the target protein, one for the E3 ligase, connected by a linker. Their molecular weight is higher than typical drugs, but they are chemically synthesized, not produced by living systems, and that is why they are small molecules. Okay, these are organic small molecule scaffolds, not peptides or antibodies. Why they are not biologics? Because they are not proteins, peptides, antibodies, nucleic acids or cell. They do not require biological production system. They function through small molecule mediated protein degradation, not biologic mechanisms like antibody binding. Makes sense. In PROTACs, in short, PROTACs are large small molecules. They sit typically between small molecules and biologics in size, but by definition they are small molecule therapeutics. So this is where I compare small molecule ATP competitive catalytic inhibitors with PROTACs. Okay, so please remember this for your exam. Okay, remember everything about PROTAC. So they are target activity dependent mechanism of tumorigenesis, target activity dependent and independent mechanisms of tumorigenesis for enzymatic sites. Okay. Usually PROTACs are much more efficient than just the thing. What does this mean here? What is the meaning of target activity dependent and independent mechanism? Can you explain to me? So keep in mind, kinases have active site by which they do phosphorylation. So if you make a small molecule inhibitor, you basically shut down the mouth of the kinase. It cannot talk, but it can still do a lot of other things, protein-protein interaction, things like that. But if you degrade the protein, the whole protein is gone. The kinase activity is gone, plus all the interactions are gone also. So that is why it is more powerful. Occupancy-driven mechanism of action works as stoichiometric. So if there are 100 molecules of kinases, you have to have at least 100 molecules of the inhibitor. But see PROTAC, they were recycled. Did you see that? Because once the protein is degraded, protein is out. See here. See, it is recycled. It just brings two people together, then it goes and brings another two people together, another two people together, another two people together. Okay. So that's what it is telling you here. You don't have to work, it can work at substoichiometric concentration, requires continuous exposure for therapeutic efficacy. This does not require continuous exposure. Binding to protein active site required, this is very good. Binding to active site not required, can target undruggable proteome. I will also show you some very cool ways of PROTACs where you don't even have to use a small molecule. We'll come to that. That's when we will call it a biologic, because we will attach a nanobody to it. Okay, what is nanobody will come later, but that's very interesting. That's when all the biologic people say, yeah, it is a biologic. Okay. Okay, specificity depends on the binding of the, do you guys understand druggable and undruggable? Everyone? Do you understand undruggable? No. That's okay, don't worry about it. Because you guys are from different background, you know, in some cases these people are strong, in some cases you guys are strong. So what happens is druggable means sometimes, druggable means there is a pocket, there is a lot of things there so that you can make a molecule which binds there. Undruggable means that it's like flat surface like this. How do you make a small molecule which will bind the flat surface, right? So that's where antibodies can be very useful. They can come and bind here. So PROTACs can be designed which can inhibit protein-protein interaction or things like that. So it can, I will show you some example that it can also target those proteins which are hard to target using just small molecules. Okay, that's what it means. Specificity depends on binding to the target. There is just one small molecule and one target, so the whole thing depends on their specificity. But here the E3 ligase is also part of the game. Drug resistance easily develops. Here it develops, but it is a lot slower because you are making the whole protein disappear. So what happens is that, suppose this is a protein that you are targeting, right? You use a kinase inhibitor and immediately it will, like Gleevec, it will develop mutation there, and then Gleevec will not work. Then you make second dasatinib, then it will develop another mutation, dasatinib will not work. Then you make third generation, it will develop another mutation, that drug will not work. But here you are making the protein disappear, so there is no mutation there, right? So eventually it will develop resistance by making some other protein instead of this protein which has disappeared. But it is relatively slower as compared to a small molecule. Okay? So delays easily develops, here it is delayed. Okay, good pharmacological properties, pharmacological profiles can be poor because sometimes the size is too big. Okay, all right. Now we come to monoclonal antibodies. You guys know about monoclonal antibodies. We'll finish it by 12 so that we can go to the next one. Okay, so antibodies are Y-shaped, produced by the immune system B cells. We have already discussed that. So usually you inject a mouse or a rabbit, you isolate the B cells, you make hybridoma, you isolate monoclonal antibodies. Don't worry about the protocol of making monoclonal antibodies. It's a very routine process for the last 50, 60, 70 years. Okay, you can directly make it in rabbit, or you go in mouse and you make a hybridoma and you isolate the antibodies. Antibodies are Y-shaped molecules which bind any protein of interest with 100% specificity. Okay, so EGFR. So see, this is EGFR is receptor tyrosine kinase. So this is the kinase domain. So if you have to use a small molecule inhibitor, you will inhibit the kinase domain here. But if you have to use antibody, how many of you have used antibody in the lab? Only you, only 2 people. So antibodies are huge molecules like 250 kilodalton. So they will not cross the cell membrane. So antibodies are used for cell surface proteins. Okay. So if you have to inhibit EGFR receptor signaling, which is a receptor tyrosine kinase, or VEGFR is for angiogenesis, it binds the outside and turns off the signaling. Signaling will not go in. If you have to use small molecule inhibitors, so this is the actual ligand. So if the ligand binds, signaling goes on. If the antibody binds, signaling is switched off and no signaling. If you use a small molecule inhibitor, you are inhibiting using the tyrosine kinase domain. Do you see the difference? So monoclonal antibody binds up, this thing does. Polyclonal antibodies are those, poly means many, right? So they bind the target at many different sites. Like they will, so this is the target, they will recognize this part, this part. These are different antibodies, so different antibodies are binding different parts, right? Monoclonal will recognize only one part of it, so they will only bind that, but they are equally effective, okay? So this is going to only recognize this part of the antigen. Yes, go ahead.
So the linkers have atoms like oxygen, which has potential of forming hydrogen bonds. The linkers.
Say that again. The linker
The
of
linker,
PROTACs,
yeah.
which have oxygen.
They have the potential of forming, say, hydrogen bonds. So will they not interact with other proteins?
So you know, when hydrogen bonds are formed, right? So what is the definition of hydrogen bonds? When hydrogen bonds form, the hydrogen which is attached to oxygen, right? So that is this hydrogen attached to more electronegative atom here, right? That's why it is partially positive charge. In this case, it is a ether linkage. So you are right, the hydrogen which is attached to a more electronegative atom can form there, but that is not very likely. But the hydrogen bond of water is forming, which is good because it is more soluble in the hydrophilic environment, so it's keeping it open. But if you replace oxygen there, so what I'm trying to say is that yes, hydrophilic water will form hydrogen bond, but not from other proteins or other things will come. So it is good if it is forming hydrogen bond with the water. So you are attaching oxygen for that purpose there. So that it is more water soluble and more exposed, like keeping the two things together. If you make just CH2, it will just fold on itself to avoid water. Okay? All right, so key advantages of monoclonal antibodies: extremely high specificity. It is as specific as it gets. Antibodies are just outstandingly specific. No offs, so if you make it against this guy, it will only bind this one, not 100,000 other proteins, extremely specific. So no off-target effects. Small molecules, off-target effects, because they are going to hit something else also. diverse mechanism of action. They can block signals. Many times what happens is that they can block signals many times because antibodies bind, immune system gets activated, immune system comes and attacks it. So not only they are stopping the signal, but just by virtue of being there, they attract immune system cells and then they will kill the tumor cells. So that's a real advantage. You can attach a drug to the antibody and then it is bound here, the drug will go inside, you will kill it. So antibodies bring a whole new bunch of things. You can add radioactivity to it, radionucleotide will go inside. Okay. They also as a delivery vehicles, and remember PD-1, PD-L1, they are blocking the signal, immune system gets activated. And now what we do, although even if we make it in mouse or anyone, we have humanized the antibody so that the antibody does not trigger our own immune system. So it seems like human antibody when it goes inside. Okay, now we know the sequence of the antibody, so we can actually generate it outside of, we can even generate it in E. coli. Okay, so what are the challenges? Extremely high cost, administration, it has to be given by IV. Some patients can react strongly against antibodies, like really the immune system will get triggered. Difficult crosses like blood-brain barrier, antibodies are not going to cross blood-brain barrier, and if somebody has brain cancer, that is not likely to work. Target cells, especially cancer cells, can still mutate or adapt, and so the antibody no longer binds there. So if antibody binds here, they will change the sequence here, antibody will not recognize. So infection risks, these patients may develop infection risks, and antibodies are large proteins, 150 kilodalton. They cannot cross the cell membrane. Okay, and if they cannot cross the cell membrane, you cannot really, so you can only target cell surface proteins. Okay, so that is where these nanobodies will come into play. So these are very famous antibodies, which are all, the red ones are very famous antibodies. These are FDA approved against different kinases, HER2, EGFR, and MET kinases. And look at that, how many, from 53 to 2023, many antibodies have been approved. These are, look at that, different antibodies. Now, very rapidly, we are increasing it. Initially, there were few of them, now so many of them are approved. Okay, now we come to nanobodies. So what is the biggest problem of antibodies? They are huge. So there is this animal called camelid. Okay, it looks like camel, but it's slightly different. Okay, so this is our antibody, which has a heavy chain and light chain, but their antibody is like this, really tiny, 15 kilodalton antibody. So instead of making human antibodies, we make nanobodies, and you can even get them across the cell, and they are very powerful, very powerful. Okay, so let's look at these nanobodies. So nanobodies are very small antibody fragments, 12 to 15 kilodalton. Those are almost 150 to 200 kilodalton. They are derived from camelid heavy chain. So those of you who do not know about antibodies very much, this is the part here. This thing you see here, this design, this is the part which will recognize the antigen. So one antibody to the second antibody will differ in this thing. So in this case, this is the pocket which will recognize the antigen. Okay, so much smaller than monoclonal antibodies, and because of their size, nanobodies can be expressed inside cells as intrabodies. Okay, nanobodies can target intracellular kinase. Of course, you express them inside the cell, they will bind, the kinases are rendered ineffective. So you can add the DNA inside the cell, and then, or you can attach to cell penetrating peptides. Do you guys know what is cell penetrating peptide? Anyone knows what is cell penetrating peptide? No, cell penetrating peptides are sequences like 10-mer sequence, arginine rich. So they will bind and they can cross the cell membrane. So if you attach something to that thing, so suppose you attach the 10-mer to any protein, the whole thing will go inside, because this has the capability of crossing the cell membrane. So you can get nanobodies inside, or you can package them in nanoparticles or liposome, and you can get inside. And once they go inside, they can bind the kinase catalytic domain, regulatory domain, phosphorylated activation loops, and block kinase activity. Basically, they can work. See, nanobodies are not druggable. So even if the surface is very straight, nanobodies can recognize this part. And once they go there, they can block kinase substrate interaction. So suppose BCR-ABL does cancer by binding this protein, the nanobody can come in between. They will not bind. They can bind the active site. They can bind to any part of the kinase. They don't depend on the pocket because this is an antibody, right? So they can stabilize inactive conformation. So they are extremely powerful. So there are many antibodies against kinases you can see here. And then you can even attach. So now PROTAC can become a biologic. So remember, one part was the ligand which was bringing, instead of ligand you add nanobody. So one side is binding the protein of interest, one side is bringing E3 ligase, it will get degraded, right? Now it becomes a biologic. Okay, so this is how they work. This is the camelid thing, okay? So this is conventional antibody, this is a nanobody. So camelid antibody are like this, and you can just cut out the pink part. This is the nanobody here. So this is camelid antibody which do not have light chain. See, this whole Y shape is heavy chain. This light blue is light chain. So you cut out this part and you make the nanobody. And you add the nanobody, it goes inside the cell. You can attach different kinds of nanobodies like this. And this one, you can attach a nanobody here, and this will bring the cancer cell together. Okay, and this is an immune cell, so they can bring two things together. So you give this nanobody, two different nanobodies attached together. One will bind the cancer cell, one will bind the immune cell. It brings together, it will kill, the immune cells will kill the cancer cells. Okay, this can use as a diagnostic thing. So advantage of nanobodies, they can access intracellular kinases. Monoclonal antibodies cannot, small molecules obviously can. Specificity is very high, unlike small molecules. Anytime you hear antibodies or nanobody, 100% specific towards the target. Small molecules are usually not. They can bind cryptic pockets. Cryptic pockets are those pockets which appear when the kinase goes active, inactive. So it's not always there. When it is changing conformation, these pockets appear, disappear. Nanobodies can just hold on to whenever they appear and bind. Okay, small molecules usually can't. Stability is very high. Monoclonal antibodies not that much. Nanobodies are very high, and so are small molecules. So small size, they can go deep inside, penetrate, high affinity, very stable at extreme pH and temperature. You can express it in E. coli, ideal for intracellular. The only thing is that nanobodies will degrade, so you have to hide it in such a way so that they don't degrade. So if you attach it to something, it can have high serum life. Okay? So nanobodies are extremely versatile. I already told you about that. So this is comparing nanobodies and small molecular inhibitors. Nanobodies are much small. This is obviously a biologic. You already know everything about organic molecules. They are very effective for extracellular. They are also effective for intracellular, provided you get them inside the cell, which is easy. Okay, and I already told you about they can target active and inactive conformation. They can also access cryptic pockets which appear, disappear. Nanobodies, okay, excellent in blocking protein-protein interactions. Small molecular inhibitors are not. So you guys have not seen these fields developing. I have seen these fields developing as I was starting my own career. That time the biggest challenge was how do you interrupt protein-protein interaction. Nanobodies can break the interaction. Small molecules are very hard to do that. Go ahead.
Nanobodies don't create immune response. They will be like an foreign body to our body, right?
Nanobodies usually don't do that because they are too small. They can, actually they are big enough to carry this thing, but nanobodies are, usually they don't actually. So maybe they also are humanized in such a way that the immune system doesn't attack, but that's a very good point. I've never heard nanobodies, but they can, so you can always humanize them. Humanize them because you may make them look more like humans. But I will check on that, that's a very good point. Whenever I read nanobodies, I've never seen immune response. But if it is there, it should be here.
It belongs to some different species, right? So it must.
But we have also cut it down from the camelid part also. We have just taken the variable part which binds.
Every domain, because this is derived from the domain from a specific domain of a like genome. It's basically genomic, right? If a human is producing light chain and it's specific to me only, but this is from different species.
See, so your point is very good. So this is our antibody, right? But our means ours and mouse and rabbits. So humans have something special about this thing. The dark blue part is the only part which is recognizing the antigen. Now here only the dark pink part is the one which is recognizing. So we are cutting out all the part of camelid here. We are just left with this pink part here. And pink part is the one which is binding that one. So it is really small. And if we know what are the things which can trigger it, we can change that. We can make it more human-like. Like there is nothing human-like in this because we don't even have this thing. But once you sequence enough nanobodies and if you see any immune response, you can figure out which part is doing it. And you can make it more human-like. Like you can do mutations to see that it doesn't trigger immune response. When we can be so smart in coupling two antibodies together to bring immune cells and those things together, we can be very smart in making it humanizing, right? That's what humans are very smart in, right? Okay. So nanobodies are in early clinical development for cancer, approved for other diseases, okay? For small molecules, of course, we have many FDA things. Small molecules can be given orally or IV, readily enter cells, simple pharmacokinetics. These are injected proteins. They are not going to cross cell membrane by themselves. So either you have to get them through nanoparticles or something. You still have to do more work to get them inside. But once you get them inside, they work. So this already we have discussed. Okay, now see, nanobodies can be attached to radionucleotide, to toxin, to fusion proteins, they can be packed in liposome. So you can attach a lot of things to nanobodies, and look at that, you can attach PROTAC to nanobody. See, and now nanobody is the one which is recognizing, so no ligand part. It is binding the protein of interest. Now this is a biologic because it has a nanobody attached to it. And once it binds there, it will degrade the protein. We are going to talk about CAR T therapy, right? So what CAR T therapy does, it recognizes it is expressed in immune cells, T cells, that's what the CAR T comes from, and it has a chimeric receptor which will bind to the tumor cells, right? So tumor part, this thing, the top part which recognizes the tumor, you can replace it with nanobody. And now it will bind the tumor cells and T cells will attack it and kill it. So this is CAR T normal cells. So normal cells, they will have, this is the nanobody one. So this nanobody is the one which will recognize tumor cells and kill it. Here, this is the receptor which will recognize the tumor cells and kill it. So you can even attach nanobody to a CAR T cells. Okay. So let's stop here now because it's been 12:09. We can have a 5-minute break and then we'll continue with the next lecture. Which lecture number? This is second. Protein kinase interaction inhibitor. So this is just to show you protein kinase. This is the PROTAC part which I already told you. I told you about monoclonal antibody. So if there are some small molecules or peptides which can break the interaction between kinase and substrate, these are protein-protein interaction inhibitors. So see, this is a kinase called pKa. So pKa, when cyclic AMP binds, this is the regulatory domain, blue part leaves and this kinase becomes active. So this is the kinase domain, and when it is bound like this, it is inactive. So when it leaves, this guy becomes active. So if you add a peptide to bind this one, so even it has left, but a peptide is bound there, it is no longer active. So that is a protein-protein interaction inhibitor. So you can use some inhibitor which will disrupt kinase binding to substrate. So this is a kinase and this is a protein substrate. So if you do not let them bind, there is no way it will phosphorylate it. So if you use a small molecule so that this can never come close to this thing, the small molecule is in between, it is not going to work. So that's what is a protein-protein interaction inhibitor. So it prevents, it blocks kinase scaffold protein, prevent kinase dimerization. So some kinases, they have to come together to dimerize before they become active. If you add an inhibitor, they will not dimerize, it will remain inactive. So that's what is the meaning. So that is your, so these are some of the drugs, okay, which are FDA approved, and they basically, they do not go in the ATP binding site, they break the interaction between the kinases. So for example, this is a drug, this tramitinib. So this is binds to MEK kinase, and when it binds to MEK kinase, it inhibits its interaction with RAF and also inhibits its interaction with ERK1, ERK2. So if the top guy is not able to transmit the signal, it is not going to activate it. Similarly, it cannot transmit the signal bottom also. So basically it's a protein-protein interaction inhibitor. That's what it is. This is BCR-ABL, the famous BCR-ABL. So look at BCR-ABL. This is the BCR part. This is, look at my structure here. So this is the kinase domain, this is the SH2, this is the SH3 domain. Kinase, SH2, SH3. I had drawn for SARC, but this is another tyrosine kinase. So this kinase, this is the kinase ATP binding site. This is where ATP is bound and substrate is phosphorylated. But it also has, this is unique to BCR-ABL, it has a myristylation site. Myristylation site is a hydrophobic site which binds a very hydrophobic tail. Okay, so what happens is that in the active state of ABL, myristoyl pocket is open, and this is ABL without the BCR part. So this see, this is the tail which is binding the SH2 domain here. Here the tail has a myristyl group which binds its own pocket here. So the kinase is inactive. Can you see that? I can see you guys are very tired, but just stay with me for 5 minutes. Okay, this will be done in less than 5 minutes. So it shuts it off. See, the whole thing is collapsed on top of each other. When BCR is there, then this is open. That's why BCR causes cancer. This does not cause cancer because this is switched off. But BCR is there. But if you make a compound, this is an FDA approved drug. If you fill in this pocket, somehow BCR collapses like this, and the kinase is shut off. So this is a protein-protein interaction inhibitor. This is not, it has nothing to do with that. The last part I have briefly told you about antibody drug conjugate. So you are giving the antibody, but you are attaching the drug to the antibody. So it's a dual thing. So this is antibody, and you have attached a payload to it, which could be a cytotoxic drug. So this is the cancer cell. Antibody binds here, it gets inside, releases the drug, kills the cancer cell. That's ADC. Okay, so these are FDA approved ADC for different kinds of cancer. You can see that. This is that drug, right? Herceptin attached to a toxic molecule. So double attack. Okay, and I promised you I'll tell you what exactly are clinical trials. I keep on saying that. So look at that. There was one time a student asked me, what are phase 2, phase 1, phase 3 clinical trials? So this is what you guys are doing in your lab. Okay, whatever the early discovery is there, that is that. Then we say, okay, this is, so we, all the way cell culture, animal, all these things are called preclinical. Then if you have a new drug, you apply for this new drug thing. Then once you apply and it is approved, then it goes to phase 1. So phase 1 has 20 to 100 participants. Okay, phase 2 has 100 to 300 participants, and phase 3 have 1,000, so more than 3,000 participants. So many drugs cross phase 1, phase 2, and then they fail in phase 3. And that is very expensive for the company. And once they pass that, then it goes through the approval process, and finally it is approved. All right, I'll stop here. This is the last slide. So let's have only 5 minutes break. Okay, I'll start in exactly 5 minutes. Okay, it's 12:15. We'll start at 12:20.